Okay, so I'm going to start talking about the special senses. We should finish this subject Tuesday, so I'm not going to go through all of it now. And since we did talk about a little bit of this in class, there may be some things that I kind of look over and let y'all review. So as we discussed, there are 5 special senses. One of the differences you may notice here, this is another word that they sometimes use, again, to mean equilibrium or balance. And if you remember, it was the vestibule of the ear that carried that information. So that's why sometimes you'll see it as vestibular sensation as well. This is really a very key word that you should note additionally. So I know with neurons, we talk about electrical signals, which are sent because ions, charged particles, are moving across a membrane. But the point is to initiate the electrical signals, they had to originate as a stimulus. So again, we're going to see how light can be converted or transduced to an electrical signal as it hits the retina, okay, or how sound can be transduced, okay, to electrical information that lets us comprehend what we're hearing. So we already talked about the differences. We got those really good. So just review that. Okay, so again, as we look here, notice that with general senses, the stimuli initiates the receptor portion. In this case, will be sent towards the neuron cell body, then down the axon portion, and of course, convert the signal to the axon, eventually carries it to the specific part of the brain. With special senses, again, except smell. Smell is simple, like general senses. The other difference you'll see, though, as well, are these neurons here. Notice that these are bipolar. Bipolar neurons are rare, and they're only found with special senses. So with smell, the receptor portion still goes straight to the neuron membrane. But for all of the other special senses, head movement, again, is going to include equilibrium, balance, vestibular function. You have a special cell for that sense, and then it sends it down the associated neuron. Okay, so yes, we're going to look at each individual special sense, but there are some generalities that if you pay attention to and you kind of understand the different steps that can occur, it makes learning how each special sense differs a lot easier. So first of all, again, except for smell, you actually have a receptor cell. And so most cases it will tend to increase the amount of neurotransmitter release. Sometimes, though, it might decrease. Regardless, what we see is there is a change onto the neuron, okay, because of the stimuli that was transduced. The neurotransmitter might be released as soon as the stimulus hits the cell. It might be released by a neuron later down in the pathway. Then you need to figure out what actually made the neurotransmitter released. Okay, was it light, like for the eye, sound for hearing? Chemicals would be when we smell or when we taste. So you need to know what the initial stimulus for each special sense would be. So just like we discussed with neurons, to change a membrane potential, which is what an electrical signal is, you have to allow ions to move. Okay, so it will either open ion channels, allowing them to move, or close, which stops the movement, so a charge could build up. So again, we're going to pay attention, okay, to each special sense. Does it maybe cause depolarization? Is it causing hyperpolarization? Okay, and just note the differences. So as we visualize this process, again, the stimulus depends on what special sense you're talking about, but the stimulus will initiate the movement of ions across the membrane. Okay, and these movement of ions are going to bind to these vesicles that contain the neurotransmitter. When the binding occurs, It forces the neurotransmitters to be exocytosed. And then, of course, we would know on the next neuron, you would have that ligand-gated channel that the neurotransmitter would bind to and then cause more ion movement in or out, okay, at the next stage. Okay, so first smell, olfaction is for smell. This is how we detect what we call odorants, which of course are different chemicals in the air. So first of all, where the olfactory epithelium is found, which are where the receptors are to distinguish smell, it's going to be in the superior portion of our nasal cavity. Okay, so we have these special olfactory neurons. Again, you'll notice special sense. So we see bipolar neurons, and they have a chemoreceptor that the chemical, the odorant, binds to. Okay, and that's going to be transduced to a electrical signal. Around these neurons, you have basal cells. These basically replace the olfactory as they get damaged. Okay, so constantly different odorants or other allergens, we have to have new cells that can respond to chemicals. And then you have supporting cells that basically help make sure that the basal and the olfactory neurons are getting the nutrients and things that they need to work properly. Okay, so we already mentioned cranial nerve. So all of these electrical signals are going to be sent down cranial nerve 1 towards the brain, okay, down the axons. These nerves basically join with the olfactory bulb. If you remember, the olfactory bulbs sit just above the cribriform plate of the ethmoid bone. Basically, you're talking about, if you can kind of palpate your frontal sinuses, about 3 inches past that very middle part of your forehead. That's that cribriform plate. If you remember, the cribriform plate had this big line in the middle. We called it the crista galli, and it kind of looked like it had little holes punched through it. Okay, the little holes are where these olfactory nerves go through to join with the olfactory bulb. Then the electrical impulses are sent down the tract to continue towards the brain, okay, so it can interpret these odorants or smells. Okay, so again, you see the neurons. Notice they're bipolar. They're going out through those tiny little holes in the cribriform plate, and they are joining neurons that are forming the olfactory bulb. What's kind of interesting here is as air is inhaled, it actually has to mix with a little water before it can appropriately bind and start this process. Okay, so how does water basically mix with the odorant? It's from the mucus, okay, that sits in our nasal cavities, protecting it and keeping it moist. Okay, so it's going to bind to these proteins, which is going to basically allow the receptors to be stimulated. And they're tiny little hairs or cilia on those neurons. Once it reaches the neuron, of course, it's transduced to the electrical signal. So for olfaction, it's not just straight the odorant, which is our stimulus here, doesn't directly affect ions to move. Okay, we have a G protein involved. So I think of G protein as go. Go as in go activate inside the cell. Okay, so once this binds, this now G activated protein moves, it goes, and it's going to bind to an enzyme. Remember, anytime things end in ASE, that's an enzyme. And it's going to speed up the process, okay, of converting ATP energy into the form, okay, that we need for our ion channel, which is cyclic AMP. Once the cyclic AMP is made, then it appropriately can move to the channel, opening it, and in this case, allowing 2 different cations to enter. So we have more sodium and calcium in higher concentrations outside of the cell. That's why both of those molecules move in. And of course, when you have positive coming in here, we are depolarizing the inside of this cell. Okay, so for each of the special senses, you're going to need to know the pathway. How do these neural signals go to the parts of the brain that actually make sense of these electrical signals? So we have axons of the olfactory neurons or nerves that carry it to the bulb. We've already talked about that. The bulb itself is actually within part of the brain. So the axon terminals are going to synapses where they drop off information with the next cells. The next cells are the mitral cells. So they go to the dendrites of the mitral cells into the bulb. Mitral cells actually form the majority of the olfactory tract, and they're going to travel between that bulb and the primary olfactory cortex in the temporal lobe. Again, remember, olfaction is one of the only things that does not first go through the thalamus before it goes to the appropriate cortical region of the cerebrum. Okay, so in this temporal lobe, and other portions of the olfactory, we're going to mention some of these structures here. This is where we are aware of what we're smelling. It's pleasant, unpleasant, and basically once you've been around as a child, you kind of learn new smells, and as an adult, you don't even have to look at what you're smelling a lot of times to be able to identify a potential odor. So notice a lot of the components here: amygdala, hippocampus, hypothalamus. These are all portions of the limbic system, which is one of the major systems in the body that controls emotions, which is why the sense of smell or olfaction is strongly linked to an emotion. So a smell of something can, trigger maybe music that you heard at the time, or maybe a scene. Okay, it triggers a memory. And it could even trigger to the point where it could cause a visceral response. What I mean by that is if it's a really bad smell, a lot of people, dead, necrotic, rotting tissue, it's kind of one of those smells that will make you vomit. It almost just has that immediate effect on a lot of people. Okay, so basically all we're seeing here is that same pathway. So these are our bipolar olfactory neurons, okay, that transduce the odorants into electrical signals. They go through the cribriform plate. They're going to join with the mitral cells. So you have a conversion of electrical impulses there down the olfactory tracts. Okay, and then they go straight to where the primary olfactory areas are. Again, they're not going to pass through the thalamus. Anosmia is a condition in where a person cannot smell. It may not be a big deal. It can be very serious because a lot of times, especially negative odors, can actually warn us of potential danger, like smoke, or if food doesn't smell good. Also, olfaction and taste are very strongly linked together. So if you cannot smell food, even when you're stuffed up, you're not going to enjoy the complex taste of food as much either. So things that can cause anosmia or hypoosmia, which is reduced. A lot of it makes sense. Things that you would naturally think of. Anything that basically is going to block air, therefore odorants, from being able to bind to the receptors. So you could have nasal polyps. Those can build up in sinus cavities if you have chronic sinusitis. If you fractured your nasal septum, of course, if you've broken your nose, which could very easily damage the septum or other bones and cause a lot of swelling, inflammation up there. Respiratory epithelium, again, any of that from, again, chronic sinusitis or cold or a virus, which in those cases, it's temporary. Okay, as soon as your stuffiness goes away, it's resolved, but that's not necessarily always the case. So when it's permanent, if you have a head injury that's damaged the neural pathway and that information can't go to that primary olfactory area, you're never going to be aware that you're smelling. So for example, concussion is temporary. Okay, so you could very easily get that back. Anything that damages the cribriform plate, which you would have to have extensive upper head injuries for that to happen. Even Alzheimer's disease, Parkinson's disease, impairs the sense of smell. And that's actually one of the signs. Parkinson's, basically the brain neurons are starting to die and move, so that one makes sense. Alzheimer's has a lot to do with memory, and again, smell and memory are very much linked. And in that case as well, you have neurons dying, which is what's causing your dementia. Hyposomnia, naturally, basically, as you age, you're not going to be able to smell quite as well. It's usually not something people are significantly aware of. Okay, as you get upper 60s, 70s, you might be a lot more aware of that, which is one reason why people's tastes change so much as they age. They can't smell as much, so they might like more stronger, different tastes. Okay, so review, you can do these on your own. Okay, so now gustatory sense, which of course is taste. So once again, with taste and smell, you're talking about chemicals, okay, or the stimuli that are binding to the receptor. So we can use the term chemoreceptors for both of those. This one is more complicated. Okay, first of all, as we already mentioned, smell does affect taste. You also have thermoreceptors, that's temperature. Nociceptors, which are basically going to give you more information about textures of food. Of course, with the actual gustatory or taste receptors as well. So you have a taste bud, which are basically just clusters of these receptor cells that are all over the tongue, and some are in your mouth, some are down the throat. Each taste bud basically is linked to one of the cranial nerves that we'll talk about that, of course, carries this information to the brain so we can interpret, okay, what we're tasting. So when someone sticks out their tongue, okay, and you see tiny little bumps, you're not looking at the taste buds. You are looking at the papillae. Okay, so papillae are large kind of protrusions where the taste buds can be found. And we have 4 different classes. We define them based on shape. So circumvallate or vallate are the largest. In the back of the tongue, they actually kind of have this V-like shape, which is kind of how they get their name. They can have up to 100 taste buds per papillae. And again, this number, you're going to see this number decrease as we age. Fungiform are more mushroom shape, hence the fungi. But they don't have near as many taste buds on them, typically 10 to 20. But those are scattered all over the tongue. Folate are on the sides. They're really only important in childhood. This is an adult that really doesn't do anything. It's more about the taste and stuff associated with milk. Filiform are the ones that do not have anything to do with gestation at all. Okay, these are detecting the other things that we basically look at when we're determining if we like a food. The texture of it, the temperature. Some people can eat cold food, it doesn't bother them. I particularly really like, if it's supposed to be warm, I want it to be warm. Certain people, they actually like the taste of strawberries or even sushi, but they can't get over the texture of it. And the filiform are what you can kind of feel as well, especially on the tongue of a cat. If a cat licks you, it can actually kind of hurt sometimes a little bit. It's because there's a lot more keratin in those. So taste buds are usually on the sides, okay, lateral portions of the papillae. So again, you basically have the same idea as what you have for smell. So anytime you see these similarities, note them instead of memorizing everything again. Okay, so you basically have the actual receptor cell itself for the special sense. You're always going to have the basal cells that will replace, okay, the older receptor cells, and then you'll always have the supporting cells. That's pretty standard. Again, we talked about this, and that we have 3 different cranial nerves that are associated with taste. So we have 7, which is facial, 9, glossopharyngeal, and 10, which is vagus. Okay, so again, you kind of see the circumvallate making that kind of V that we talked about. The fungiform are everywhere. It doesn't really show the filiform because they don't actually have taste, but they would be all scattered throughout here as well. And again, this other picture, you see, again, the lateral sides are where you see those taste buds. And basically, for us to actually be able to taste, again, the chemical, okay, the part of the food, has to dissolve a little bit in saliva, and it needs to enter the little taste pore. Okay, and you have little cilia in this little taste pore, and at that point, it can be transduced to electrical signals. So us being able to determine or identify if we like a food or what it is relies on the detection of actually only 5 classes of chemicals. So it's the mixing of these different classes that how we can get really particular about maybe dark chocolate versus milk chocolate versus white chocolate. So sweet taste, of course, you're going to have sugars, simple sugars, glucose, fructose. Bad things, lead paint, ethylene glycol have a sweet taste. People might continue to consume it because of that, and those, of course, are poisonous. Sour taste will have hydrogen ions associated with it. So a lot of times you're going to see acid. Of course, acids are a measure of hydrogen. So the more acidic, the more hydrogen, the more of the sour taste that you'll have. So citrus, lime, lemons will have the taste to them. Salty is by metal ions. Sodium, of course, sodium chloride is salt. Potassium as well. Bitter flavors are anything that that has basically nitrogen in it. Bitter and sour are different. A lot of truly bitter things can be poisonous and harmful to the body. Therefore, if things are too bitter, we do not like them. Most people do not tolerate things being too bitter. Sour is different. Umami is basically what we call tasty. It's associated with meat or broth, something very flavorful, usually produced by glutamate or other amino acids. A lot of things that contain MSG kind of have that umami taste to them. Okay, so of course, there are those of us that aren't too particular when we eat, and other people that are highly particular. We say they have very sensitive palates. A lot of these people are chefs. So what they really are is they're a super taster. So basically, super tasters, the fungiform papillae, which only have a few taste buds, they have more, and theirs are more sensitive. So if something is slightly sour or has more complex layering of flavors, they're going to be able to determine that. This is kind of funny. Look of a person who would be a super taster, and how they can figure this out. One way is they can use this one chemical. It's really not important the name, but for short we just call it prop. And most people taste nothing, about half. It's a little bitter, and then of course the super tasters, okay, the people with really good palates would just make this face and want to get it out of their mouth immediately. And of course we do see that it changes, okay, as we age as well. A lot of things too, which is important to note, preferences occur while you're growing up. So of course, everyone thinks their own mom's cookies or whatever tastes the best because that's what you're used to. And a lot of that is also triggering those memories of good, happy times that you're associating with that specific taste. This is just if you're kind of interested in the soup taster, you can read a little bit more about it. Okay, so the physiology of gustation. So again, we're going to have a stimulus just like with olfaction. It's going to be chemical. It's going to bind to, instead of cilia, this time we have other little hairs, microvilli, okay, on the gustatory hairs. And basically, like I said earlier, you have to have water again involved. That chemical, whether it's a metal or an amino acid or fructose, has to dissolve in saliva. So if your mouth is super, super dry, you might notice that you really can't taste stuff. That's why. Okay, pathway. Again, for all of the special senses, you must know the pathway. Okay, so again, as we see, about the anterior 2/3 of the tongue is by the facial nerve. The posterior 1/3 is glossopharyngeal. Pretty much almost everything else is vagus. Okay, again, you'll see, you're going to notice that these neurons are going to leave. They're going to make their way to the brain. First, they're going to go through the brain stem. So in the medulla oblongata, the specific nuclei, they're called solitary nuclei. It's basically going to go through. It's going to go up from the medulla through the pons here, through, as most everything else, through the thalamus, where it will then go to the primary gustatory cortex, which of course is, some books say parietal lobe, other books will say the deep insula, which is basically kind of right there. Your book obviously uses parietal. Okay, and you can review these on your own. Although you probably won't see these exactly, you could see something very similar on tunics, your exam. Okay, so now we're going to talk about the eye, which of course is what I spent the majority of class time today talking about. And I only put this one up. I took out all the other slides that have this. This is lab only. So keep in mind that you are responsible for basic anatomy structures like the cornea, the lens, the vascular tunic, the ciliary body, the few things we talked about in class. And again, you will talk about those in lab. So those of you that might not be taking lab, I would make sure that you know basic anatomy as well. So again, the only reason I included this lab only one on here was for this little guy right here. So LR stands for lateral rectus. So the lateral rectus is controlled by cranial nerve 6, which is abducens. Superior oblique is controlled by 4, which is trochlear. All the other, we have 4 more extrinsic or external eye muscles. Those are all controlled or regulated by the oculomotor nerve, which is cranial nerve 3. So there are 6 extrinsic muscles of the eye. Okay, so these are going to control looking up, down, cross eyes, laterally. And remember, there's 2 internal or intrinsic muscles of the eye. We talked about both of those in class. That was the iris and the ciliary body. Okay, we already discussed these layers. So again, know the layers, know the major structures found within each layer, and if they have basic functions, make sure that you know those as well. And it says these help maintain the shape. I should go ahead and tell you, the one that really helps maintain the shape the most is going to be the fibrous layer. The white, the sclera, has a lot of collagen fibers, and this also has good attachment for those extrinsic muscles of the eye. Okay, so just review basically the anatomy. If you know all of the terms on this sheet, you should be good. Okay, and just really, really basic stuff about what it does. So again, we talked about these things. Okay, so again, since I talked about some of this stuff, I'm going to kind of don't want to just keep going over. We'll point out one thing here. So again, the vascular tunic, that's the middle tunic here. Basically, once you get back past this part, this is all choroid. These 2 are those intrinsic or internal smooth muscles of the eye that I talked about. So remember, smooth muscles are going to be controlled by the autonomic nervous system versus somatic, where the extrinsic muscles are controlled. Breeze over this. Okay. I don't actually know if I mentioned the word pupil, but everyone knows the black hole that you see, it is actually an opening. It's the pupil. So the black is actually from the choroid that's inside of the eye. And something else that I should have pointed out is that the choroid and part of the retina, they both have melanin in them. So think about if you're wearing black on a sunny day, that absorbs light. The purpose of those is to absorb light in one spot to the back of the retina and not have light basically bouncing around everywhere inside of your eye. So notice you basically have 2 smooth muscles that are around here. The more internal layer here is the sphincter muscle. So when that contracts, it will make the pupil smaller. When the dilator muscle contracts, it will actually allow the pupil to increase in size, therefore reducing the amount of light that's entering the eye.